In this series of videos, we will take a deep dive into the development of antidepressants. While learning about the history of 3 drugs, you will also exercise big themes related to scientific discovery and drug development. If you are a pre-med college student, an AP biology high schooler, or simply someone with some background in biology who's interested in learning more about depression and science, this series is for you. Years of research in the science of learning have demonstrated that deep engagement with the materials is the best way to learn and retain information. So try not to just watch this video. Take some notes, write some questions, engage in the activities, and truly be here. And with that in mind, in today's video, we will investigate experimental findings from basic and applied research leading up to the development of a drug, dissect the steps in the FDA drug approval pipeline, explain the connections between antidepressant classes and mechanism of action, evaluate the implications of communicating scientific discoveries to the general public. I'd like to start us off with a curious short video.
Brother and sister, together we'll make it through. Oh yeah. Someday a spirit will take you and guide you there. I know you've been hurting, but I've been waiting to be there for you. And I'll be there just helping you out whenever I can.
Okay, let's pause here because I have a few questions. What do these people have in common? Why are they looking up dramatically? And why was that girl wearing shoes in her bed? Let's just see where this goes.
Everybody's free. Everybody's free. Oh, yeah. To feel good. Oh, to feel good.
Nothing, I repeat, nothing could have prepared me for that Earth spins into a Prozac pill ending. This 90s masterpiece of special effects was an advertisement aired in TVs all over the US. Jokes aside, a close look at this advertisement for Prozac can help us understand what this drug meant in the market of antidepressants. The message is that anyone can benefit from Prozac, from a mother experiencing postpartum depression to a teenager having melancholic thoughts. Brothers and sisters, as the song says, are all free thanks to Prozac. It's very interesting that Prozac is a prescription drug, but its marketing is targeting prospective patients instead of the doctors directly. And that says a lot about what the drug meant for the first time you had a prescription medication being discussed outside of clinics and informal conversations. The massive promotion of Eli Lilly's drug paid off. Prozac was named one of the pharmaceutical products of the century by Forbes. And in 2002, it had reached 40 million users worldwide. It made it to pop songs, movies, books, and even magazine covers. What's the source of this success? Is it on the catchy name or on a very massive marketing campaign? There surely isn't a single answer to this question, but there's reasons to believe we can start by looking at the star molecule behind all this frenzy. Meet fluoxetine, the active compound in Prozac. Fluoxetine is often described in terms of its market success, and that is a totally valid interpretation. After all, at some point, Prozac alone was responsible for 2/3 of the revenue at Eli Lilly. But the motivation to produce a drug like Prozac wasn't entirely monetary in the first place. A 1975 market analysis had indicated very little value in antidepressant sales. According to them, the market was satisfied with MAOIs and TCAs. Remarkably, and perhaps against all odds, including a lot of skepticism in the field and many twists and turns, a research team at Eli Lilly Laboratories decided to pursue an effort to create a drug that could selectively inhibit serotonin at the synaptic cleft. But how did they get to serotonin in the first place? In our last video, we talked about the monoamine hypothesis of depression, which sustains the idea that a depletion in the levels of monoamines in the brain was the underlying mechanism of depression. This hypothesis was further strengthened by the clinical value that TCAs and monoamines added to the market because they work by either inhibiting the reuptake of monoamines or inhibiting their oxidation. Around that time, chapters in neurochemistry textbooks characterized the processes of serotonergic neurotransmission, from synthesis to reuptake and degradation. Importantly, histochemical studies had increased the confidence in neuronal fibers specific to serotonin, making it a promising candidate for targeted treatment. With this knowledge, a research group at Lilly Laboratories became interested in the reuptake of serotonin as a potential drug target for antidepressants. Unbeknown to them at the time, another team was working on a similar question. In 1970, Brian Molloy and Robert Rathbun were trying to identify chemical agents with the therapeutic activity of TCAs, but fewer undesirable side effects. They thought this could be accomplished by inhibiting the reuptake of only one type of monoamine, in other words, selective inhibition. For that, they developed analogs of diphenhydramine, an antihistaminic that displayed monoamine uptake inhibition. Using an in vivo paradigm called reversible hypothermia, they found LY94939 was as effective as TCAs in inhibiting monoamines. So they tested that substance in 3 related molecules for how well they could inhibit noradrenaline uptake in comparison with the diphenhydramine. LY94939 was as good as the diphenhydramine in inhibiting noradrenaline uptake, but worse at inhibiting serotonin uptake, hinting at some specificity in its inhibitory activity. Having a substance as a selective noradrenaline uptake inhibitor, they thought that its analogs could also demonstrate selective inhibition. So they decided to test how well these drugs and all its analogs could inhibit serotonin and noradrenaline uptake in rat brains using synaptosomal preparations. In this test, they found that fluoxetine oxalate and norfluoxetine were the most potent inhibitors of serotonin uptake and weaker inhibitors of noradrenaline uptake. Simultaneous with testing the selective inhibition of these other compounds in vitro, they became interested in developing a methodology that could see how well fluoxetine could do in vivo. Very conveniently, Fuller had already studied the effects of different compounds on monoamine oxidase. He found that p-chloramphetamine caused serotonin depletion in the rat brain. This effect was shown to be blocked in vivo by drugs that inhibited serotonin uptake. So now they could use p-chloramphetamine to test how well the drugs that were inhibiting the reuptake in vitro would do in vivo models. They found that fluoxetine oxalate was the most effective, but it was too hydrophobic, and if you remember from our first video, this is often a problem for oral drugs. So they requested that the chemists Molloy and Hauser prepare the hydrochloride salt version, which was comparatively more polar. After running many studies, they confirmed that fluoxetine did preserve selective inhibition in vivo, something that tricyclic antidepressants couldn't do, because it had active metabolites that would actively inhibit both noradrenaline and serotonin. These findings solidified fluoxetine as the first selective inhibitor of serotonin reuptake in vivo. Okay, I think we covered some ground on the experiment so far. Let's recap. Monoamine oxidase inhibitors and tricyclics are the known classes of antidepressants. It seems that the market is mostly satisfied, but they have many undesirable side effects, which makes them treatment alternatives only for more serious cases of depression. Otherwise, psychotherapy was still the main go-to in clinical practice. Based on the efficacy of monoamine oxidase inhibitors and the known role of serotonin as a neurotransmitter involved in the mood, the group at Eli Lilly Company became interested in developing a molecule that could selectively target the reuptake of serotonin. They found one molecule that demonstrated selective inhibition of serotonin uptake in vitro and in vivo using rat models, and that molecule is fluoxetine. Is this convincing enough for you? What other questions would you have about fluoxetine at this point? Take a minute to pause the video and jot down 3 questions you would like to answer next. I don't know where your questions went, but I like to think of this in terms of safety and efficacy. You will see that in many steps of the drug pipeline, there are 2 recurring questions. Is this safe? And is this effective? Having initially answered these two questions, at least in rat models, it feels like the natural next question is to understand how exactly the drug works. Asking this question of how can help us lift this veil of ignorance and actually look at the pathways at which the drug interferes and potentially undesirable side effects. So slowly what we're trying to do here is approach a more clear picture of the mechanism of action for fluoxetine. Fluoxetine decreased the conversion of isotope-labeled tryptophan into serotonin and 5-HIAA in the rat brain, most evidently in the cerebral cortex and the brainstem. But it had no effect on the accumulation of tryptophan or in the activity of tryptophan hydroxylase, the enzyme that catalyzes the hydroxylation of tryptophan into serotonin. At this moment I had to stop and wonder for a second. Because if fluoxetine isn't actually changing any of the steps in the synthesis of serotonin, what is it doing? And that's where the beauty of metabolic pathways shines. Because these synthetic pathways aren't always working at the same rate. This network of reactions is responsive to factors that either downregulate or upregulate them. Let's take a look at the synaptic cleft once again. This concentration of serotonin essentially controls how much will be synthesized by the presynaptic cell. Normally, this works in a feedback that maintains balance. When there is too little serotonin in the synapse, the receptors upregulate the synthesis of serotonin. When there is too much, the receptors are silenced and the synthesis of serotonin is downregulated. However, when fluoxetine comes into the scene, it inhibits the reuptake of serotonin to the presynaptic neuron. When serotonin accumulates in the synaptic cleft, the receptors tell the cell to decrease the production of serotonin. This felt very counterintuitive to me. How come fluoxetine is increasing the availability of serotonin when it's actually decreasing its synthesis? Maybe you already figured this out. Although fluoxetine does decrease the synthesis of serotonin, it increases the concentration of serotonin in the synapse. And that's where a neurotransmitter shines, right? And this is where serotonin's main role happens. It activates the postsynaptic neuron. So the net result is increased serotonergic activation, because serotonin has a prolonged presence in the synapse. Fluoxetine is considered the first SSRI, or selective serotonin reuptake inhibitor. And now that we've talked about all these reactions, can you actually dissect this acronym and explain what each letter means? The findings from the research groups of Eli Lilly were presented at the annual meetings of the Federation of American Societies for Experimental Biology and the American Society of Pharmacology and Experimental Therapeutics in 1974. But they were met with a lot of skepticism in the field, especially because fluoxetine didn't show a significant effect on available models of depression, such as the force-swim test. This well-established research paradigm was widely used to test preclinical antidepressant activity. It consists in placing a rodent, often a mouse or rat, into a cylinder full of water, deep enough that its paws can't touch the bottom, forcing the animal to swim. A healthy animal will move a lot initially, trying to escape, until it reaches immobility and moves just enough to keep its nose above water. When exposed to the antidepressants available at the time, tricyclics and monoamine oxidase inhibitors, the time spent in immobility compared to movement was reduced. The problem was, the force-swim test didn't show a significant effect for fluoxetine. As you may imagine, this test, or any other attempt of modeling a condition like in vitro or in vivo testing, will eventually fall short. Although they serve a very important function in safety and efficacy testing, they can't have the final word about how a drug will work in human metabolism. Thus, after completing toxicological evaluation in rats and dogs, fluoxetine moved to the next step in the drug pipeline, clinical data. Fellow human beings, I present to you the FDA approval process in a nutshell. Every promising young drug has a sponsor, a company, a research institution, a startup. With preclinical data from animal testing at hand and a plan for human testing, they file an IND, Investigational New Drug Application. This application is reviewed by the FDA and an institutional review board to ensure that it's safe to proceed with tests in humans. The role of the IRB is to evaluate all the details going into the trial protocols, inclusion and exclusion criteria, medications and doses, study goals, length of study, criteria for termination. Only then the sponsor can start phase 1 studies. These are trials conducted with a sample of 20 to 80 healthy volunteers to answer questions about safety. What are the most common side effects, how the drug is metabolized? If phase 1 studies demonstrate acceptable side effects, phase 2 studies kick in. These trials typically have a sample of a few dozen to a few hundred volunteers who have the condition to be treated. Although safety is still considered, the main focus is on effectiveness. Does the drug help treat the symptoms? If phase 2 shows effectiveness, often in comparison with a placebo or the standard treatment, the FDA and the sponsor meet to determine the protocols for phase 3. These typically have the largest samples, from several hundred to a few thousand subjects. In this phase, they can identify less common side effects and determine effectiveness on different populations. Typically, the sponsor and the FDA meet again to discuss post-market requirements upon approval. Only then, the sponsor files a new drug application, the formal step of asking for the FDA's approval to market a drug in the US. While the team at Eli Lilly waited for FDA approval, a group led by Carlson at Astra Pharmaceutical introduced another SSRI to the European market. Perhaps luckily for Eli Lilly, the European competitor was shortly retired from the market due to an unanticipated side effect. In this scenario, Fluoxetine finally got a green light from the FDA in December 29, 1987, debuting in American markets as the first SSRI. Dr. Wong, who was part of the team from the earlier experiments to the approval, said that the news brought not only excitement but also an ultimate vindication, as we had carried the burden of the project and had been ridiculed for many years as the ones who discovered and developed a molecule looking for a disease. I can't imagine how rewarding it must be to see the work of 16 years unfold into a drug that brought new hope to millions of patients around the world. The arrival of such a safe treatment had a big implication for clinical practice. It shifted the cost-benefit analysis for clinical practitioners. For older antidepressants like MAOIs, the associated cost with undesirable side effects made it such that they were only prescribed for more serious cases of depression. For these patients, the clinical benefits would outweigh the risks. However, with a drug like Fluoxetine, with milder side effects and minimal risks of drug-drug interactions, the costs are diminished. By comparison, the benefit was worth it even for milder cases of depression, which now could be treated with a drug prescription. In fact, Fluoxetine remains one of the safest and most tolerable antidepressants. On the one hand, the popularization of Prozac helped destigmatize depression. A lot of marketing in the 1990s centered around the idea that antidepressants worked by fixing a chemical imbalance of neurotransmitters in the brain. This biological perspective on depression helped counteract the common misconception that individuals have control over their condition. They can just try harder and be happier. As you can imagine, it lifted an enormous weight off of the shoulders of people struggling with depression. But this wasn't an all-positive shift. Characterizing depression as only a chemical imbalance is a gross misrepresentation, and this idea was never really pursued in academia. Yet, it gained a lot of traction in shaping the public's understanding of depression. A study with undergraduate students in 2007 found that almost 85% of participants believed that chemical imbalances cause depression. Note here that although Fluoxetine and many other antidepressants were developed under the assumption that increasing the availability of serotonin can improve symptoms of depression, this doesn't mean that a lack of serotonin is causing depression. What it means is exactly that. This mechanism has a demonstrated clinical improvement, as supported by a strong body of evidence that shows that antidepressants work. Around 60% of patients respond to treatment with SSRIs, observing a significant improvement in symptoms. In academia, they're working with the biopsychosocial model of depression. This mouthful is trying to reflect the complexity of factors that interact in causing depression. Growing evidence shows that elements in biology, individual psychology, and social contexts can increase the risk of depression. Although the exact mechanisms are still being studied, we do know that they each have a sometimes small, sometimes big influence on the etiology of depression. It turns out that a simplistic view of a complex mental disorder is a disservice to approaching the epidemic of depression. When we don't appreciate the complexity of this disorder, we can easily lose sight of the many possibilities for intervention surrounding mental health. We covered a lot of ground today. Let's recap. The knowledge accumulated from the first class of antidepressants and basic findings in neurochemistry inspired the Pharmacological Research Division at Eli Lilly to investigate serotonergic pathways as a target for antidepressants. In a series of experiments in vitro and in vivo, they arrived at fluoxetine hydrochloride, a molecule that selectively inhibits the reuptake of serotonin at the synaptic cleft. Despite skepticism, fluoxetine successfully passed a long pipeline of preclinical and clinical evidence required by the FDA, inaugurating the class of SSRIs. Under the brand name of Prozac, fluoxetine remains one of the safest and most financially successful endeavors of the pharmaceutical industry. Its massive but questionable marketing strategy contributed to the public's oversimplification of depression under the idea of chemical imbalance, in contrast with the psychosocial model of depression, mostly accepted in academia. You can find all the references we used to produce this video in the description. There, you can also find additional materials that complement the topics we talked about today. See you in our next video.